Right, this is chapter 24, part A, on the urinary system. So the kidneys are kind of like the body's water filtration system. So they're the major excretory organ, and they're going to help to maintain the homeostasis, or that stable internal environment of the body, by regulating our water volume as well as solute concentrations in that water. So ion concentrations in the extracellular fluid, things like sodium, potassium, calcium. Ensures long-term acid-base balance, excretes metabolic waste, toxins, drugs. Another function of the kidneys that we talked about in the endocrine system chapter was the production of erythropoietin, that's going to regulate red blood cell production, as well as the production of renin. So the renin was that enzyme that's going to kickstart that renin-angiotensin-aldosterone mechanism for blood pressure regulation. So the kidneys are the major organ of the urinary system. The urinary system also includes the ureters, which are just going to transport the urine formed from the kidneys and carry it to the urinary bladder. The urinary bladder is just that temporary storage for the urine, and then the urethra is what's going to help transport the urine outside of the body. So kidney location and external anatomy. So they are retroperitoneal in the superior lumbar region. Right? So just a little bit in the lower back behind the peritoneum. So you notice the right kidney is a little bit lower than the left because we have the liver up here that's going to kind of crowd and push that kidney down a little bit. And we've already talked about the adrenal glands in the endocrine chapter, so they sit just atop each kidney there. So they have a convex lateral surface, so it's kind of a broader curve, and then the concave medial surface, the smaller curve, is going to be the location of the renal hilum. So this will be where the ureters, blood vessels, nerves, lymphatic vessels will enter and exit the kidney. So there's 3 layers of supportive tissue around each kidney just to help maintain its overall structure and suspend it in the proper location in the abdominal cavity. So it has the renal fascia, which is the outermost layer. So it's a dense fibrous connective tissue that's going to help to anchor it in place. Also has a perirenal fat capsule. You see kind of this little bit of fat around the kidneys. So this is just going to serve kind of as some extra cushioning and support. And then the fibrous capsule is going to line the actual surface of the kidney, and its function is to help prevent spread of infection. So internal anatomy of the kidney has 3 distinct regions. So the renal cortex is the outer portion, and the renal medulla is the mid portion. So the medulla is going to contain the medullary pyramids. Right? So we have kind of these pyramid-shaped sections of medulla here. So the papilla are going to be the tips of the pyramid. So this would be where the urine is going to be expelled from. Okay. So the renal pyramids you see are separated by renal columns. So the renal columns are just extensions of this cortex. It's going to kind of go toward the center of the kidney. And then the lobes, the renal lobe is just a medullary pyramid and its surrounding cortical tissue. So generally there's about 8 lobes per kidney. So this whole thing would be 1 lobe. This whole thing would be a lobe. Here's our 3rd lobe. Right, so the pyramid and the renal columns beside it make up 1 lobe of the kidney. So the 3rd region is the renal pelvis. So this kind of funnel-shaped tube here that's going to be continuous with our ureter. So the renal pelvis is made up of the minor calyx, or calyces, plural. So this is just the tips near the tips of those pyramids, those papilla, that the It's going to collect the urine draining from the renal pyramid papilla. Right from the tips of those pyramids, urine's going to come out into the minor calyces, and then the major calyces are going to collect urine from the minor calyces. Right? So we have a minor calyx here, just kind of at the tip, and then where these all kind of merge together, it's going to form the major calyx. So urine flow after its formation is going to go from the renal pyramid into the minor calyx, which drains into our major calyx, and then the renal pelvis and out through the ureter. So we said the function of the kidneys was to filter the body's blood supply. So since the kidneys are going to clean the blood and help adjust its composition, it has to have a very rich blood supply. So it's a very vascular organ. So the renal arteries are going to deliver about a quarter of the cardiac output to kidneys each minute. So nephrons are the structural and functional units that form the urine. There's roughly 1 million of these structures per kidney, and they consist of two main parts. So the renal corpuscle, shown here, and then the renal tubules coming off of that corpuscle. So there's two parts to the renal corpuscle. So the first part is the glomerulus. So the glomerulus is just a little ball or tuft of capillaries that have a highly fenestrated epithelium. So remember, fenestrated just means it's very porous. So it has lots of these little holes or windows in them. So this is going to allow for efficient filtrate formation. So the filtrate is just the fluid that comes from the blood plasma that the tubules are going to process in the formation of urine. Right? So we have different ions and chemicals and things in the blood that we need to filter out and cross through these capillary membranes. The second part to the renal corpuscle is the glomerular capsule. So sometimes called the Bowman's cup or the Bowman's capsule. It's just a little cup-shaped or hollow structure that's just going to help surround and support and protect this little ball of capillaries in the glomerulus. So we said the second part of the nephron were the renal tubules. So in total, it's only about 3 centimeters or about 1.2 inches long. But the three major regions or parts of the renal tubules, so you have the proximal convoluted tubule. So proximal meaning that it's closest to that renal corpuscle or closest to the point of origin. We talked about the anatomical terms. The nephron loop, so it just kind of comes down and loops up and around. And then finally the distal convoluted tubule. So this one's farther from the corpuscle or that point of origin. So then from here, these distal tubules are going to drain and empty into collecting ducts. So looking at our proximal convoluted tubule, so it's composed of some cuboidal cells. It has a very dense microvilli layer to form the brush border. So this is just to help increase the surface area. So when we're secreting and reabsorbing substances, we need a lot of surface area to kind of maximize the efficiency. So functions of your proximal tubule include reabsorption and secretion, but these are generally going to be confined to the cortex. So this is trying to show the kind of the separating line between the cortex and the medulla. So the nephron loop, sometimes referred to as the loop of Henle. So it's just this little U-shaped loop. It consists of two limbs. So we have a descending limb that's going to go down or descend. Right? And then the ascending limb. So it's going to ascend back up toward that distal tubule in the cortex. And the distal convoluted tubule, similar to the proximal, it has those cuboidal cells, but it has very few microvilli. Okay? So it's going to function more in secretion than absorption. So the microvilli increased our surface area in the proximal tubule so we can absorb more ions and chemicals and things. So in the distal tubule, since it's primarily concerned with just secretion, instead of absorption, it doesn't really need all that extra surface area from the microvilli. And it's also going to be confined to this cortex region. Right, so then we said the distal tubule is going to empty the filtrate into the collecting duct. So collecting duct can receive filtrate from several different nephrons. Right? So then the collecting ducts are going to run down through the medullary pyramids. So when you look at the pyramids, it's what's going to give them this kind of striped appearance here. So then the ducts will fuse together as it reaches toward the tips of the pyramids at the papillae to deliver that urine into the minor calyxes. So the renal tubules are associated with two types of capillary beds. So we've already talked about the glomerulus, right, which is just like a little ball or tuft of capillaries. And then we also have peritubular capillaries, so shown in the purple here. So we have some small little capillaries that are going to kind of interweave around these tubules. Right? And some types of nephrons called juxtamedullary nephrons. So they're just ones that run a little bit deeper into the medulla. They have a special type of associated capillary bed called the vasa recta, but it's going to function pretty much the same, serve the same purpose as our peritubular capillaries. So looking at the glomerulus, we said was just kind of a little tuft or ball of capillaries in the nephron. So these capillaries are specialized for their role in filtration. Right? So we said they're highly fenestrated. They have lots of these pores or little windows, right, in them. But they're different from other capillary beds in that they are fed and drained by an arteriole, right? So most capillary beds, right, they are fed, the blood arrives to the capillary through the arteriole, but then it's going to drain the capillary in the venule. Right? But because we're not dealing with gas exchange or anything like that, it's going to also be drained by pretty much that same arteriole. Okay? So the afferent arteriole is going to enter the glomerulus, right? So A for arriving, right? And the efferent arteriole is going to leave the glomerulus, so E for exiting. Okay. So then from there, the efferent would feed into either those peritubular capillaries or our vasa recta capillaries. Right? So the blood pressure within the glomerulus is fairly high for a couple of reasons. So one being that our afferent arteriole has a much larger diameter than the efferent arteriole. So we have a higher flow of blood or a faster flow of blood into the bed, but then we're going to narrow that efferent arteriole, so it's going to slow it down a little bit. Right? So these arterioles are high resistant vessels. So when we decrease the size or diameter of the arteriole, that's going to slow down the blood flow. So think if we go from 4-lane highway down to a 2-lane, right? So traffic's going to slow down and kind of get backed up. But the high pressure here is necessary to help push the solutes and things across that filtration membrane, right? So we can filter the blood. So looking at the peritubular capillaries, right? So again, peri means like beside or next to. So these are capillaries that are beside or next to the tubules. Right? So these have a fairly low pressure, very porous capillaries that are specially adapted for the absorption of water and solutes. So they're going to rise and come off of these efferent arterioles, and they're just going to kind of cling to the adjacent tubules throughout the cortex and empty into our venules. So each nephron also has what's called the juxtaglomerular complex, or sometimes it's referred to as the juxtaglomerular apparatus, or we'll just say the JGA, right, JGC. So it consists of some modified portions of that distal end of the ascending limb. Right? So we have our ascending limb coming up. So then it's going to make contact with that afferent arteriole. Right? So the tubule that is leaving the nephron, so after everything's kind of been processed throughout the tubules, then it's going to make contact with the blood that is arriving to the nephron. Right? So they're going to kind of check in on each other on certain solute concentrations. Right? So we'll talk a little bit more about that in a bit. But primarily it's important for regulating the rate of filtrate formation and blood pressure. Right? So we're kind of doing a double check on our filtrate that we've made and its composition right before we continue. So there's three types of cells found within the juxtaglomerular complex. So the macula densa cells here are part of that ascending limb of the nephron loop, right? So shown here in the purple here around the edge. Right? So these are going to contain some chemoreceptors that are going to monitor the sodium chloride or the salt content of the filtrate to make sure we're not losing too much sodium or absorbing too much sodium. The granular cells, sometimes called juxtaglomerular or JG cells, are smooth muscle that are found in this arteriole, in our afferent arteriole. So these are going to act kind of as mechano or stretch receptors. So similar to how we had those baroreceptors in the carotid arteries to monitor blood pressure, we have some of these mechanoreceptor stretch recepting cells in this afferent arteriole, so we can make sure that blood pressure isn't too high or too low. So these are the cells that will actually release that enzyme renin to kickstart your renin-angiotensin-aldosterone mechanism. Right? So if blood pressure gets too low, right? So since the kidneys are constantly filtering the blood, so they're also monitoring our blood pressure. So blood pressure gets too low, these granular cells or JG cells of the afferent arteriole will detect that and release the renin, which would kickstart our aldosterone mechanism to increase blood pressure. And then the third type of cells found in the JGC complex are the extraglomerular mesangial cells. So these cells that are kind of just in this space in between the arterioles and the nephron loop or the tubule, right, that fill this space. So located between the arterioles and the tubule cells, and they're interconnected with gap junctions. So meaning that they're able to pass ions and chemical and electrical signals to one another more quickly. Right? So they can also pass signals to these macula densa cells and the granular cells. So they have a connection to both types of cells that they can contact with. So looking at kidney physiology, so we said that the kidneys are highly vascular. They have a very rich blood supply because they're going to filter roughly a quarter of your blood every hour or so. So roughly 180 liters of fluid will be processed daily, but of that, only about 1.5 liters would actually form urine. Right? So the kidneys are going to filter your entire plasma volume at least 60 times each day. Right? And because they are working very hard, they're also going to consume a lot of oxygen. So 20 to 25% of total oxygen is going to be used by the kidneys doing this job. Right? So the filtrate is basically that blood plasma fluid minus any proteins and things that are too large to pass through the filtration membrane. Right? So then urine is going to be produced from that filtrate. Right? So about less than 1% of the original filtrate containing your metabolic waste and any unneeded substances and toxins and things like that. So there's three processes involved in the formation of urine, as well as adjusting our blood composition. So step one would be glomerular filtration. So this would just be filtering the fluid portion out of the blood. So pretty much everything except cells and proteins will be filtered out through this membrane. So fluids, ions, chemicals, all of that will pass through here. So the cells and proteins are just too large to pass through our filtration membrane, so they're going to remain in the blood. Tubular reabsorption. So we're going to selectively return roughly 99% of the substances that are filtered from the blood and return them to the blood. So we reabsorb things that we want to keep, right? So we don't want to pass through all of our sodium, all of our potassium. We want to hang on to most of it that we need. Right? So we're only going to secrete the excess. So this is showing number 2 for tubular reabsorption. So it's reabsorbing back into the bloodstream. Okay. So then the third step would be tubular secretion. So we're going to secrete anything that we want to remove from the body. Right? So we'll secrete from the blood or filtrate to the tubules, collecting ducts to go on to form urine. So the process of glomerular filtration is actually a passive process, so meaning it doesn't require any energy on our part. So it's going to be powered by the hydrostatic pressure of the fluids going through that filtration membrane. Okay? And also there's no real reabsorption into these capillaries at this point. So remember we said it was a fairly high pressure type of capillary. So normally in your typical capillary beds you want a very low pressure so we don't rupture or damage those kind of fragile, small, delicate vessels. But in the kidneys, a higher pressure is required in order to kind of force these fluids and solutes across this membrane. Okay? So, and again, how we get that high pressure is by having a large diameter afferent arteriole and a smaller diameter efferent arteriole. So we're going to slow down the flow and have a little bit of backup or traffic jam in here. So we increase the pressure, kind of pushing things across this membrane. So the filtration membrane is the porous membrane between the blood and the interior of our glomerular capsule. So we have our blood capillary and then the membrane from our glomerulus capsule. Okay? So things like water and solutes that are typically smaller than your normal plasma proteins will be able to fit through these spaces in the membrane and be filtered out and come through. But like I said, normally cells that are too large would not be able to fit through this membrane and pass through. Okay. But 3 layers of the filtration membrane. So you have your fenestrated epithelium, right, of the glomerular capillary. So again, fenestrated just means it has these little pores or fenestrations. Right? So remember, fenestra means window in Latin. So we have lots of little windows or pores in the membrane of the cell or the capillary. Okay. The basement membrane is going to be the fused basal lamina of the blood vessel and the glomerular capsule. Right, so it's kind of the glue that's going to hold these two together. And then the third layer are the foot processes of the podocytes. Right? So pod, podo, I think maybe foot. So these are foot cells. So they have these foot processes, right? Or just these little like extensions that are going to interweave and kind of cover and like a mesh over the capillary. Right? So we have these filtration slits. So we form kind of just another layer of our filtration membrane. Okay? So it's got to pass through the fenestrations or the pores in the capillary, and then it also has to pass through and fit through these filtration slits of the podocytes. Right. So the outward pressures that are kind of influencing and promoting our filtrate formation would be the hydrostatic pressure and the glomerular capillaries. So it's essentially the blood pressure within this little ball of capillaries. Right. So that's going to be our primary force that's pushing these solutes and fluids out of the blood. And again, it's a fairly high pressure compared to our normal capillary beds. Like I said, we would want a low pressure in a typical capillary bed because the vessels are so small and delicate. But in the glomerulus, they're a little more sturdy and can handle a higher pressure. Right? And again, the reason that the pressure is so much higher is because the arterioles are different diameters. So our afferent arteriole has a larger diameter, and then the efferent has a smaller diameter. So it's going to slow down and increase the resistance to the flow, right, once it enters into the glomerulus. Right. So there's also some opposing inward pressures that we have to contend with that are wanting to inhibit or work against our filtrate formation. So one of those is the hydrostatic pressure in the capsular space. So it's just the pressure of the fluid within the capsule. Right. So arrows going this way are inhibiting our filtrate formation. Right. So we had our glomerular capillary pressure, the HPGC here, right? So it's promoting solutes pushed out, 55 millimeters mercury. So hydrostatic pressure in the capsular space is the fluid in here that's kind of pressing against, working against that glomerular capillary pressure. And then there's also a colloid osmotic pressure of the capillaries, or the OPGC. So the proteins in the blood that are too large to pass the membrane, they're still going to have kind of a pull or an attractive force of fluids. Right? So that's roughly about 30 millimeters mercury. Okay? Just kind of the draw or the pull of those blood proteins. Okay? So then net filtration pressure would be just the sum of these forces. So we have 55 millimeters of mercury forcing outward, right? Minus 30, minus 15, right? So that gives us a net filtration pressure of 10 millimeters of mercury. Right, so this is the actual pressure that's going to be responsible for that filtrate formation. Right? So later on we talk about the glomerular filtration rate, so how much filtrate the glomerulus is making, so what's the rate, how fast is it making this filtrate. We can control this or kind of manipulate it by changing our net filtration pressures. So the glomerular filtration rate, or GFR, is just the volume of filtrate formed per minute by both kidneys. So it's kind of like the cardiac output of the kidneys. So basically the filtrate output, filtrate formation by both kidneys per minute. Okay? So it's going to be directly proportional to that net filtration pressure. And so that's kind of the primary force pushing out those solutes and fluids. So if we were to increase our net filtration pressure, then that's going to increase the pressure or the force that's pushing those solutes and water out of the blood. It's also proportional to the surface area available for filtration. So those mesangial cells that have smooth muscle, they're able to contract or dilate to increase or decrease how much area or space we have for filtration. And then filtration membrane permeability. So these capillaries are much more permeable than other capillaries because they have a role in filtration. So looking at regulation of the glomerular filtration rate. So generally a constant rate of glomerular filtrate formation is important to allow the kidneys to make the filtrate and maintain homeostasis. So the kidneys have a couple of intrinsic or auto-regulatory mechanisms to help maintain that filtration rate. Also, filtration rate can affect our blood pressure. So an increase in glomerular filtration rate is going to cause an increase in your urine output, which would lower your blood volume, which lowers blood pressure, right? And vice versa. So if we have a decreased urine output, it means we have more fluid in the blood and our blood pressure is going to be higher. So this would be more the goal of your extrinsic controls to help maintain systemic blood pressure. So we've already talked about some of these in some previous chapters, some ways that the nervous system and the endocrine system can work to help maintain our blood pressure. Okay. So looking at the intrinsic control, so intrinsic meaning contained, self-contained within the kidneys themselves. So it's auto-regulation. They regulate themselves with no outside assistance. So it's pretty good at maintaining GFR at a nearly constant rate. So if our mean arterial pressure gets outside normal homeostatic ranges, our blood pressure, then those auto-regulatory mechanisms might stop being as effective and we would have to tap into those extrinsic mechanisms. But your two types of renal auto-regulatory mechanisms are the myogenic mechanism and the tubuloglomerular feedback mechanism. Right? So essentially with the myogenic, myo meaning muscle, so we are either dilating or constricting that smooth muscle in the renal blood vessels. So local smooth muscle contracts when it's stretched. So an increase in the blood pressure is going to cause those muscle cells to stretch. So they're going to respond to that by constricting the afferent arterioles. So blood pressure is normally higher in the glomerulus, but we still don't want it to get too high. So if it starts to get too high, we'll have to slow down and restrict the blood flow in there to protect those glomerular capillaries from becoming damaged by that high blood pressure. Vice versa, a decrease in blood pressure would cause a dilation of the arteriole. So if we have low blood pressure, we don't have enough pressure pushing the blood through that filtration membrane, we'll have to open up the blood vessels to allow more blood flow into that glomerulus. But both of these are just going to help to maintain our normal filtration rate despite everyday normal fluctuations in blood pressure. So here, you're looking at the myogenic intrinsic mechanism. So we have a decrease in blood pressure in our afferent arterioles. So that means that we're going to have a decrease in our filtration rate, right? So we detect that because there's less stretch of the smooth muscle in those blood vessel walls. So we respond by dilating those arterioles. So we can increase the blood flow to the kidneys, to the glomerulus, and thereby increase our filtration rate. Right, so the tubuloglomerular feedback mechanism is a flow-dependent mechanism that's maintained by these macula densa cells, right, of the JGA or JGC. Right? So like I said, they're going to monitor the filtrate sodium chloride concentration. Right? So if our filtration rate increases, that means that the filtrate formation and flow is also going to increase. So the faster the flow, that means that we have less time to reabsorb certain ions and nutrients and chemicals and things that we want to hang on to. So if it just flows right through, right, we may not be able to reclaim or recapture everything that we want to keep in our blood. Okay? So this would cause higher levels of that sodium chloride in the filtrate, right, because we're not able to absorb it adequately. So this feedback mechanism, just remember these cells are kind of monitoring the content of sodium chloride from the distal tubule, right? So it's already been processed. At this point we should have already absorbed pretty much everything that we want to keep, right? So we do one final check here with these juxtaglomerular apparatus cells to monitor our sodium chloride concentration, right? So if we haven't reabsorbed enough sodium, then our sodium chloride levels will be higher. Right? So that's going to cause the afferent arteriole to constrict. So this means we need to slow down the flow. We need to give the nephron more time to reabsorb or secrete whatever it needs to. Okay? So when we constrict the arteriole, we're slowing down the blood flow, so we're going to lower our filtration rate, but it's going to allow us more time to reabsorb that sodium that we need to. Okay? And then vice versa for a decrease in the filtration rate. So if we don't have enough sodium chloride, right? So maybe we're absorbing too much. So then instead of constricting the arteriole, we would dilate the arteriole to increase the blood flow and the filtration rate. So we can increase our sodium chloride levels. Yeah, so this example, so we have a decrease in filtration rate, which is going to lead to a decrease in our sodium chloride concentration. Right? So macula densa cells of the kidneys are going to detect this, so they're going to monitor our sodium content and cause the vasodilation. So we're going to increase the blood flow to those capillaries to increase our glomerular filtration rate. So with your extrinsic controls, this would be things that are outside the kidneys themselves. So we have some neural mechanisms as well as hormonal. So some of these we've already talked about before, so this should just be kind of like review. So the main purpose of our extrinsic controls is to help regulate and maintain our blood pressure, right? Systemic blood pressure. So generally, that's going to take precedence over the intrinsic controls. Right, so extrinsic controls will kind of override those intrinsic controls if the blood volume needs to be increased. So if we need to raise blood pressure to by increasing the blood volume, then these extrinsic mechanisms would kick in. So your sympathetic nervous system, under normal conditions at rest, right, you're not undergoing a fight or flight situation, so your sympathetic nervous system isn't really activated. So when you're just resting, your renal blood vessels are usually dilated, so we have plenty of blood flowing to those that glomerular capillaries, and the autoregulatory, those intrinsic mechanisms, are usually pretty good at handling any slight fluctuations. So under our abnormal conditions, though, so extremely low extracellular volume, low blood pressure, right? So it's beyond the point that our intrinsic controls can't fix it. So then we need to tap into our extrinsic controls. So things like norepinephrine can be released from the sympathetic nervous system, and epinephrine released from the medulla, the adrenal medulla. So they have that fight or flight response. So you'll have vasoconstriction to increase blood pressure. We can also constrict the afferent arterioles. So we can decrease the glomerular filtration rate. And as a result, the blood volume and the pressure are going to increase. And then finally, the renin-angiotensin-aldosterone mechanism that we've talked about before. So this is our main mechanism for raising blood pressure, right? Extrinsic mechanism. So those granular cells in that juxtaglomerular complex in the kidney, right? So remember those smooth muscle cells, they have mechanoreceptors to detect stretch and pressure. So if blood pressure gets too low, right, they're not going to stretch as much. So they're going to release renin. Okay, so renin kind of kickstarts this whole cascade effect. So then we end up with angiotensin II. So angiotensin II is just kind of the intermediate or the precursor. So angiotensin II is going to work for vasoconstriction to help increase our blood pressure, kind of temporarily until aldosterone can kick in and take effect. So angiotensin also stimulates the adrenal glands to release aldosterone. Right? And we know from before that aldosterone stimulates the kidneys to increase sodium reabsorption. Right? So water generally is going to follow sodium wherever it goes. So if we reabsorb more sodium, we're also going to reabsorb more water. So as a result, our blood volume increases and our blood pressure increases as well.